Welcome CSIR NET aspirants, viewers and subscribers of Biotechnica to CSIR NET Life Science Unit 13 Important PYQ with Solution Series. I'm Dr. Tanushri, and in this video I will be discussing previous year questions of CSIR NET Life Science exam based on topic molecular markers. Molecular markers, a topic you study in several units of CSIR NET Life Science syllabus, like Unit 8, which is Inheritance Biology, Unit 12, which is Applied Biology, and Unit 13, which is Methods in Biology. So let's first understand what are markers, and then we look at the type of questions asked in the CSIR NET exam based on this topic. Markers. Markers are of several kinds. Markers can be morphological, biochemical, cytological, and molecular or DNA-based markers. During the early history of plant and animal breeding, the markers used mainly included visible traits. So, morphological markers are usually visual indicators of phenotypically differing characters, such as color, shape and size of flowers, seeds or leaves, type of development of plants, inflorescences or root system, pigmentation or habit in animals. So here these two pictures shows the cows. So we can conclude that these two cows are not same as they differ in pigmentation. So morphological markers generally represent genetic polymorphisms which are easily identified and manipulated. Cytological markers are related to variation in chromosome number, shape, size and bending patterns. In other words, it refers to chromosomal banding pattern produced by different strains. So several stains like Giemsa stain is used to produce the chromosomal banding pattern. We call it as G-banding. And many more stains are available. Then biochemical markers such as isozymes and proteins. As we all know, proteins are product of gene action. The product of gene can be used as a marker for the presence of a gene. Different alleles of a gene produce different proteins. So proteins can be used as a biochemical marker, or we can say enzymes can be used as a biochemical marker. Like for example, the enzymes derived from pancreatic acinar cells, like amylase, lipase and proenzyme trypsinogen. So these enzymes have been tested as biochemical markers of acute pancreatitis, that is inflammation of the pancreas. And serum amylase specifically is the most commonly used of these in clinical practice. Then isozymes are alternative forms or structural variants of an enzyme that have different molecular weights and electrophoretic mobility, but have the same catalytic activity or function. So even isozymes can be used as a biochemical marker. Then DNA markers or molecular markers. They are defined as a fragment of DNA revealing mutations or variations which can be used to detect polymorphism between different genotypes or alleles of a gene for a particular sequence of DNA in a population or gene pool. Simply speaking, DNA or molecular marker is a small region of DNA sequence showing polymorphism between different individuals. So no two individuals have the same genetic makeup. Maybe in one individual there is a deletion in a gene occurring, but in another it is not occurring, or insertion happening or substitution happening. So we can detect the polymorphism, the differences between the individuals by using DNA or molecular markers. So we can find out the differences at the genetic level. And DNA markers or molecular markers can be hybridization based, where you use a probe which can bind complementary to the target sequence. And the example of this kind of marker is RFLP, that is restriction fragment length polymorphism. Then we have PCR based markers also. So they are based on PCR. And the examples are RAPD, random amplified polymorphic DNA. We have amplified fragment length polymorphism, that is AFLP, ISSR, SCAR, that is sequence characterized amplified region, SSR, simple sequence repeat, SRAP. And even we have DNA sequence based markers like single nucleotide polymorphism, that is SNP. Then we have diversity array technology and NGS, that is next generation sequencing. Now, in the CSIR NET Life Science exam, we can expect different type of questions on this topic. Question asked can be direct type of question. That is, they'll give you several characteristics or features of a marker and you have to identify what marker it is. Is it RFLP or AFLP or RAPD and more. Similarly, they can give you match the following type of question. So in match the following type of question, they'll give you two columns. In one column, they'll mention the different markers. And in another column, they can ask you whether the marker is multi-allelic or biallelic, or is it a codominant marker or dominant marker. So this concept, codominant and dominant, is very much important, as you will find several questions in previous year question papers of CSIR NET exam based on this concept. Then you can also expect figure-based question where they give you a band profile and they'll ask you whether the progeny belongs, which progeny belongs to the parents, or whether the marker is again like codominant or dominant. By looking at the band profile, you have to find out. And even you can expect combination-based question. So combination-based question, that is from the concept of genetics and the markers. And even in a same question, they can mention several type of markers, like they can write down the features or characteristics of several markers, and you have to find out that which statement is correct or incorrect. So statement based questions also you can expect. So let's have a look at the questions asked in previous year CSIR NET Life Science exam. Question is given below are two figures, X and Y, representing the segregation of molecular markers and their profile in parental lines P1 and P2 and F1 progeny. Following statements describe the nature or probable identity of markers in the above figure. So statements are: A. Panel X represents a codominant marker. B. Panel X represents a dominant marker while panel represents a codominant marker. So statement A and B are contradictory in terms of panel X. So statement A says panel X is a codominant marker, whereas statement B says panel X is a dominant marker. Then C statement is panel X could be SSR marker while panel Y could be an RAPD marker. And D says that panel Y represents a dominant marker, which is contradictory to what is being given in statement B for panel Y. Now you have to find out which one of the following options represents a combination of all correct statements. Now to answer this question, you need to be familiar with the concept of codominant and dominant marker. So this particular concept, as I mentioned earlier, is very, very important. Usually questions are framed on this concept. Terminology should be clear. Also, you need to be familiar with what are SSR marker, RAPD marker, and even like some other marker can be mentioned like RFLP, FLP, SNP. So you should be familiar with the all type of markers. And also you need to learn the strategy of analyzing the band profile. So let's first understand what are codominant and dominant markers. Now, not only you need to be familiar with the terms like codominant and dominant markers, but also you need to be familiar with the basic concepts of genetics as well to answer such questions. You need to be familiar with what is homozygous condition, what is heterozygous condition. So in genetics, we study that an individual inherits gene from parents, and one copy of a gene we inherit from a father and one copy of a gene we inherit from a mother. Now, there are different variants form of a gene can be present at a locus. We call them as alleles. So, either the individual can possess same allele at a locus. So, we call it as homozygous condition, as shown over here for this A and B. Or an individual can possess two different alleles at a locus, as shown for the progeny F1. So here in these band profile, you can see parent 1 is having the homozygous condition, and like both are A, but one we are writing in uppercase and another we are writing in lowercase. So usually the uppercase, the allele, when we are writing in uppercase form, we represent it as a dominant allele. So dominant allele is, or the concept of dominance is that when the two different alleles are present in a genotype, then only the trait of dominant allele is observed in the phenotype. Or you can say only the trait of one allele is observed in the phenotype and we call it as a dominant allele. So here we can say that this allele is dominant over this and this allele is dominant over this one. So this is homozygous dominant condition and this is homozygous recessive condition. Similarly for B gene, this is homozygous dominant and this is homozygous recessive. Now when an individual possesses two different alleles at a locus, we call it as a heterozygous condition. Now I gave you the concept of dominance, so it is the Mendelian concept of dominance, but there is a concept of codominance also. So in codominance, the two alleles may be simultaneously expressed when both are present, rather than one fully determining the phenotype. So I'll give you the example of both. Like for example, when we study about the dominance, so let's say when we are crossing the plant with tall stem height with dwarf stem height, so progeny will be heterozygous and you will see the phenotype of the dominant allele, that is tall. So all the progeny will be tall, will have the tall stem height. So here this allele is dominant over the recessive allele, that is for dwarf. And the example of codominance is blood group, like AB blood group. So in AB blood group, both the alleles, they equally express. So when we use codominant marker, in heterozygous individuals, both alleles are visibly expressed, which the individual has inherited from parents. So here no allele dominate the another, both are equally expressed. So here you can see the F1 is the progeny and it inherit this allele from P1 and this allele from P2, and therefore we are getting two bands in F1. On the other hand, in case of dominant markers, the heterozygous individual, so as you can see in F1 progeny, this is the heterozygous individual, only you will see one band. So only you get a band for dominant allele and not for recessive allele. Now if I don't show you the genotype, by simply looking at the band, you cannot differentiate whether it's a homozygous condition or heterozygous condition, because we are getting one band in P1 also and one band in F1 also. So dominant markers, they do not offer clear discrimination between homozygotes and heterozygotes. On the other hand, codominant markers, they clearly discriminate between homozygotes and heterozygotes. And the example of codominant markers are single nucleotide polymorphism or microsatellites, whereas the example of the dominant markers include RFLPs, that is restriction fragment length polymorphisms. So here in this question, we are given two panels, panel X and panel Y. And please note this question is asked in 2023 December evening shift, CSIR NET Life Science exam. So if we look at the panel X, we are getting, like for two parental lines, P1 and P2, we can see that they display one band each. And as we all know, progeny inherits the gene from parents only. So two bands similar to P1 and P2 is observed for F1 progeny. So we can say that panel X represent codominant marker and not dominant marker. So statement A is correct and statement B is incorrect because it says panel X represent dominant marker, whereas it is a codominant marker. Then if we look at the panel Y, it shows one band in P1 and F1. So no band is observed for P2. Therefore, we can say panel Y represents a dominant marker and not codominant marker. So as in statement D, it is mentioned panel Y represents a dominant marker, it is correct. And statement B, as I have already told you, it is incorrect. So it is incorrect for panel Y also. Then statement C is panel X could be SSR, that is simple sequence repeat marker, while panel Y could be RAPD, that is random amplified polymorphic DNA marker. Now, you should know about these markers. You should know what kind of marker is SSR and RAPD. So we know that SSR are codominant markers, whereas RAPD display dominant pattern of inheritance. So this is simply, you have to memorize the concept that you have studied, while studying SSR and RAPD, you have studied that what kind of markers they are. So that concept you have to recall here. And simply by recalling that, you can easily answer this statement whether it is correct or incorrect. So yes, panel X as it display codominance, and as panel Y it display the dominant marker. So statement C is correct. SSR is codominant marker and RAPD is a dominant marker. And as A, C and D combination is given in option 3. So answer here is option 3. Next question is, which one of the following statements regarding molecular markers for genotyping is incorrect? Now to answer such question, you have to go through each statement one by one. So statement 1 is polymorphism in intronic regions of a gene cannot be used for trait mapping. Statement 2 is codominant molecular markers can be used to detect heterozygosity. Statement 3 is sequence tagged microsatellite sites and simple sequence repeat polymorphisms are based on polymorphisms in repetitive DNA sequences. And fourth statement is RFLPs, that is restriction fragment length polymorphisms, and SSRs, that is simple sequence repeats, are multi-allelic markers. Now to answer this question, you need to be familiar with what are STMs, what are SSRPs, what are RFLPs and SSRs, what are codominant markers. So it's a memory based question and you simply have to recall about what you have studied about these markers, and accordingly you have to find out whether the statement is correct or incorrect. So if we look at the statement 1, it is polymorphism in intronic regions of a gene cannot be used for trait mapping. So what are intronic regions? Intronic regions are non-coding regions of a gene located between exons, which are the coding regions that contain the instructions for protein synthesis. Now, while intronic regions do not directly code for proteins, but they can still play important regulatory roles in gene expression and function. They can contain regulatory elements like enhancers, silencers, and splice sites that influence the expression and processing of the gene. So polymorphism, which are variations in DNA sequences, can occur in both coding and non-coding regions of a gene, including intronic regions. Some intronic polymorphism can even have functional consequences by affecting gene regulation, alternative splicing, or other processes that impact the phenotype or the trait of interest. So when we perform genome-wide association studies and trait mapping studies, researchers, they identify genetic variations associated with specific trait or diseases. So although the coding variants, like having nonsense mutations or missense mutations, can have more direct impact on protein structure and function, but non-coding variants, including intronic polymorphism, can also be informative. Therefore, statement 1 is wrong. It is incorrect because polymorphism in intronic regions of a gene can indeed be used for trait mapping in certain cases. Then statement 2 is codominant molecular markers can be used to detect heterozygosity. So as explained earlier, yes, codominant markers, they help in differentiating between homozygotes and heterozygotes, which dominant markers do not. Therefore, statement 2 is correct. Then statement 3 is that sequence tagged microsatellite sites and simple sequence repeat polymorphisms are based on polymorphisms in repetitive DNA sequences. So you should know, to answer this statement whether it is correct or incorrect, you should know what are STMs and SSRPs. So polymorphic loci present in nuclear DNA and organelle DNA that consist of repeating units of 1 to 10 base pairs and most typically 2 to 3 base pair in length are called simple sequence repeats. Simple sequence tag macrosatellite sites or simple sequence repeat polymorphisms. So microsatellite or simple sequence repeats are molecular markers based on tandem repeats of short sequences around 1 to 10 base pairs that are abundant and widely dispersed throughout most eukaryotic genomes. Like for example, the sequence can be CCTGC. Now this sequence of 5 base pair can be repeated several times. CCTGC, CCTGC, and so on. So different individuals can have this repeated several times. So there is no change in the sequence, but number of times the sequence is repeated, it can vary in different individuals. So yes, we can say STMS and SSRPs are based on polymorphisms and repetitive DNA sequences, and hence this statement 3 is correct. Then statement 4 is restriction fragment length polymorphisms, that is RFLPs, and simple sequence repeats, that is SSRs, are multiallelic markers. What is multiallelic? When we talk about multiallelic site, it is a specific locus in a genome that contains 3 or more observed allele. Like if you count 1 as a reference, then you can say it allows for 2 or more variant alleles. So yes, RFLPs and SSRs have more than 2 variants, that is they are multiallelic markers, and therefore statement 4 is also correct. Now as we have to choose the incorrect statement, so only statement 1 is incorrect and statement 2, 3 and 4 are correct. So answer here is statement 1. Another question is, mini satellites are used as marker for identifying individuals via DNA fingerprinting, as the alleles may differ in number of repeats. From the southern blot shown below, identify the progeny A, B, C and D for the given parents where M is mother and F is father. Now to answer such type of questions, you need to be familiar with what are mini satellites, what is DNA fingerprinting technique, and even you need to be familiar with the technique southern blotting. So let's first understand what are mini satellites. Variable number tandem repeats, also called as mini satellites, are among the families of repetitive DNA dispersed in the genome. So as shown over here, this is a myoglobin gene, and over here you can see this is the sequence 33 base pair, which is repeated 4 times in tandem. So mini satellites, they are repetitive DNA where certain DNA motifs ranging in length of 10 to 60 base pair are repeated multiple times, around 5 to 50 times. So if we look at this gene in individual A, B and C, so there is no change in the sequence, but the number of times the sequence is repeated in these individuals, it can differ. Like in individual A, it can differ in 15 times, in individual B it can repeat it 12 times, or in individual C it is repeated 25 times. So when we will analyze this mini satellite, or when we analyze these repetitive sequences in tandem, the profile, the DNA profile we will get, it will show the polymorphism in these individuals. They will show the difference in the variation at the genetic level in these individuals. So this way we can study the polymorphism. Now in the question, DNA fingerprinting is also mentioned, and even southern blotting. The term DNA fingerprinting was introduced for mini satellites, though DNA fingerprinting is now used in a more general way to refer to a DNA-based assay to uniquely identify individuals. And DNA fingerprinting is more commonly being used in forensic cases and even to establish parenthood, like who is the biological father or mother of a child. All you need to do is extract the DNA from the sample, which can be a blood sample or semen or saliva and other samples. So you can extract the DNA and you can perform restriction digestion by using restriction enzymes. So after restriction digestion, you will get several DNA fragments of different sizes. You can separate these fragments by performing electrophoresis, and then you can transfer the DNA from the gel to the nitrocellulose membrane. This process is called as blotting. So southern blot technique is used here. And finally, once the DNA is transferred from the gel to the membrane, you can perform hybridization. So in this process, we use the probes which are complementary to the DNA sequence, and these probes we label with a radioisotope. So on performing the hybridization, the sequences which are complementary, the probe which are complementary to the target DNA, it will bind, and then you can perform washing to remove excess of probes, and then you can perform autoradiography and can visualize the band profile. So for example, if we want to determine who is the biological father of a baby, then here you can see from the two person 1 and person 2, which are involved in the study, the blood sample is collected and DNA is extracted, and after performing the DNA fingerprinting, we'll get the band profile. Now on analyzing the band profile, we can see that band 4 and band 8 and 9 is unique to person 1 and band 7 is unique to person 2. Now we have to determine who is the biological father of a baby. So we look for the bands which are common, because many satellites have Mendelian inheritance. So some of the bands detected in individual will be common with those of mother and others will with those of father. So here out of the 4 bands, 4, 7, 8 and 9, 4th and 8th band. So this is the 4th band and the 8th band from person 1 are also present in the baby. Hence person 1 is the father of the baby. So here also in this question, a band profile, a southern blot is given to us. So all you need to do is you have to simply look for the progeny. So if the child belongs to the mother and father, their band will match with that of bands for mother and father. So if we look at if we compare progeny A, B, C and D, so for A, this band is common with mother and this band is common with father. Then for B, this band is common with father and this band is common with mother. For C, this band is common with mother and also this band is common with mother. Then for D, this band is common with father, but this band is common with mother. So if a child belongs to this mother and father, that is a couple, they should inherit the gene from both the parents and not from one of the parent. So we can say that the C doesn't belong to this mother and father, and as we have to find out the progeny belong to this couple, so the answer is option 2, which includes A, B and D, as A, B and D have bands in common with mother and father both. So we have discussed 3 types of questions from this topic, molecular markers, and you can see the questions asked are not very difficult and they are very scoring too. All you need to do is learn and revise frequently about different type of molecular markers and practice as many questions as possible. Thank you.